This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Let's see a few practical uses of all the concepts of quantitative genetics that we have learned so far. First, how can we estimate genotypic and environmental variance from an experimental design? The figure you see on the top represents the distribution of flowering timing in two wheat purebred lines. The middle line is the timing of the F1 offspring resulting from the cross of the two purebred lines, and the bottom line is the flowering timing of the F2 offspring resulting from the reproduction of F1 with themselves. For each population of parental lines F1 and F2 hybrids, we have the estimates of their mean and phenotypic variance. The parental lines are purebred, meaning that they're completely homozygous. All individuals have the exact same genotype with no genetic variation within a parental line. In this case, the phenotypic variation we observe for each parent is equivalent to environmental variation. The F1 offspring are hybrids and have no genetic variation between individuals as well, because each individual has inherited the exact same genes from the two parental lines. So once again, all phenotypic variation we observe is due to environmental variation in the F1. The F2 offspring, however, shows high genetic variability because of the recombination, random segregation of the chromosomes during meiosis, and random pairing of chromosomes during fertilization. So their phenotypic variance will be the sum of genetic and environmental variance. If we know all this, and if we suppose that the environmental variance is the same for all four groups, parents F1 and F2, because they were grown in the same field, then we can first calculate the mean environmental variance as the average of two parental and the F1 phenotypic variance. Then we can estimate the genetic variance of the F2 generation by simply subtracting the environmental from the total phenotypic variance of the F2. We obtain a result of 11.92 for genetic variance. This result is expressed in days square because variance has the statistical property to be expressed in squared units. This value is quite high, especially given that the genetic variance in the parental lines and in the F1 hybrid was 0. So it shows how effective is the recombination of chromosomes in the F1 offspring for producing new genotypes in the F2 generation. Next, how can we use heritability to predict the outcome of artificial selection? This is again the phenotypic distribution of carrot weight in a farmer's field with a mean of around 150 grams per carrot, which is already pretty big. The farmer already knows that the narrow sense heritability of the population is 0.8 and sets a selection threshold of 180 grams. This means that if a carrot weighs more than 180 grams, its seeds will be collected and then sown in the field to do the yield of the next season. So what will be the average carrot weight in the next season? The farmer does not need to wait for the carrots to grow in order to make a prediction about it, because they have all the information they need to make an estimate. The response to selection is proportional to the trait heritability and the selection intensity. Note that the response to selection needs to be calculated with selection intensity, which is simply the difference between population mean and the selection threshold. That is 30 grams of difference between what we currently observed in the field and the threshold that is applied by the farmer for selection. Given the heritability of the population, which is simply the responsiveness of the trait carrot weight to selection, the average weight increase after selection, or the response to selection, is going to be 24 grams. Now to calculate the population mean after selection, simply add the response to selection to the current population mean and you get an average carrot weight on 174 grams after selection. This is obviously an average estimate and will also depend on the environment in which the carrots will be grown. And finally, heritability can be used to make a prediction about the phenotype of an offspring in a given population if we know the phenotypes of the parents. Let's get back to the flowering timing in wheat. We know that both parental lines have average flowering dates of about 56 and 72 days. To make this estimate, we will also need information about the average flowering time of the population, and in this case, let's say that this is 58 days. And finally, let's say that we know the heritability of the flowering timing, which is 0.61. There is a simple formula to calculate what will be the average phenotype of the F1 offspring. Do not be bewildered by all the math, we'll dismantle it step by step. The mean expected phenotype of the F1 plants is simply the population mean, which we already know, plus the parental contribution to the phenotype through their heritable additive effects. Now, the parental contribution is obviously proportional to the trait heritability and to the mid-parent value, or that is the average phenotype deviation from the population mean. So here is the mid-parent value, and this is the deviation from the population mean. So after substituting all the numerical values, we obtain a mid-parent value of about 64 days. That means that if we calculate the average flowering date of the two parents, it will be of 64 days. Then we subtract this mid-parent value from the overall population mean, and we get about 6 days. This means that the flowering of each of the parents on average is delayed by 6 days compared to the population mean. Then we multiply this deviation from the mean with heritability, and we obtain an average additive contribution of the two parents to the phenotype of the offspring, which is of about 4 units or 3.79. Based on this, we can conclude that the average flowering date of the F1 offspring will be of about 62 days. So you can go back into the first few minutes of this video and check whether this result matches the values that were given there.